Hi everybody, welcome to another Chats with Children. I hope you're safe and well. It's a glorious Monday morning here in Surrey, and I'm delighted to be joined by Wai-Chi Man, who is the Product Marketing Manager for IOSP Chromatography and Mass Spectrometry at Thermo Fisher Scientific. And today we are going to be discussing ion chromatography in pharma and biopharma. So Wai, lovely to see you. How are you?
I'm fine, absolutely fine, and really happy to be chatting about ion chromatography because, I look forward to chatting to people about ion chromatography because there's not that many people out there in my local area.
Right. Well, where are you calling in from, Wai?
I'm calling in from Stevenage in Hertfordshire, UK.
All right. Well, hopefully sun's shining with you. And I know you are a former South Londoner, just like me. So it's good to see another South Londoner here on an interview today. So lovely to see you.
Excellent.
Now,
I
before
will draw in my South London accent.
well, I might start dropping my vowels and stuff when I start talking to you as well. So anyway, we shall see how that goes. Before we start talking about ion chromatography, perhaps you can give people a very quick background on your sort of experience.
Right, so my background is ion chromatography, but obviously I'm actually an analytical chemist for about 35 years, but I just choose to specialize in ion chromatography because I love all the gadgets.
Right. Okay. Well, this leads nicely on to let's start talking about ion chromatography. If I get my teeth in. So, what is a really basic question? What's the basic difference between ion chromatography and HPLC chromatography?
So basically, a lot of people would sort of say, but isn't ion chromatography liquid chromatography? Which it is. But in ion chromatography, we look at ionic species. So anions, cations, anything with a charge. And we usually analyze it in acids or base using a peak flow system. And then on top of that, a lot of the time we use conductivity as a detection, but we can actually use other things like electrochemistry to look at electroactive species as well. So there's a lot of things you can do with ion chromatography. You can link it with UV vis, add a mass spec, even add an ICP to look at metals. So
Right.
it's quite versatile.
Right. Okay. Well, as we're talking about pharma and biopharma, how can ion chromatography be used in vaccine testing?
So in vaccines, you have this thing called adjuvant, which is actually a salt. So to actually analyze that, you would actually have to analyze anions and cations. So ion chromatography is basically the technique you would use to look at those. And adjuvant helps to increase the immune response of the vaccine. So that is used quite often in vaccines. So you can actually use it for that. And also look at degradations of vaccines where it sort of degrades into sort of smaller carbohydrate impurities, whereby it's very difficult to actually analyze because it doesn't have chromophore and it's not very sort of volatile either. So it's very difficult to look at it using standard, LC methods and GC methods. So if you use ion chromatography, we are looking at the electroactive species of the carbohydrate. So you can use that to monitor sort of the degradation of vaccines.
Right. Okay. So my next question is, what is HPAE-PAD and how can it fit into the analysis of glycans?
So that that stands for high pressure anion exchange pulse amperometry detection. So it's a form of electrochemistry. So to do that, we use that to look at electroactive species. So anything that has lone pair electrons that we can reduce, or we can sort of get it to oxidize, we can actually look at those species. So it's very well used in carbohydrates. The problem is with carbohydrate analysis is that, we've got no chromophore. It's not very volatile. And so it's very difficult to analyze it. And a majority of the time, we would have to derivatize it to add the chromophore or add something onto it so that you can actually see it. But using HPAE-PAD, we don't need to. There's no derivatization. So you can just shoot the sample down in the system and it will analyze it looking at it. So this then makes it very relevant when it comes to glycan analysis, because glycans can actually sort of have bits of carbohydrates that actually fall off, or sorry, it breaks down to it and forms impurities of it. So you would be looking at sort of monosaccharides to oligosaccharides. So it's a large range of carbohydrates that you're looking for. So it's very tricky to look at those, but if you look at them, you get the information and you can actually use it to work out the degradation of your glycans. And also using that information, you can actually make out what the structure is. So for instance, at the moment, the big thing is about the spike proteins that we all talk about. Well, those are made of glycans. Okay, so the spike protein are sort of all these sort of little glycans slot together to form those. So if they break off, then obviously it changes the structure. Right. So you want to be able to analyze them.
Fine. And how important is picking the right detection?
So if you don't pick the right detection, it's quite difficult to see some of the impurities. For instance, if I say, if there's an impurity that is a genotoxin, okay, and as I mentioned before, if you've got no chromophore and it's not volatile, you can't use standard HPLC or GC to look at them. So you've got to find another way of looking at them. So if you don't have the right detection, you won't be able to see it. So then if you're not seeing it, if the drug or the vaccine or sort of, the product goes out there, somebody takes it, what happens? They could sort of like have serious injuries.
Right, showing adverse reactions. Right, okay.
Yeah.
So when a lot of people, when they think about ion chromatography in pharma, think it's just for Counterion analysis. Is that true?
The cations.
Right.
Or you're talking about counterions, yeah?
Oh, Counterion. Yes, see, I can't even I pronounced it incorrectly. Counterion, see, there you go.
Yeah, counterions, which are the
well, Counterions for me, actually, obviously, that's how I like to say them.
But what it is, counterions, it's a bit like your hay fever tablets. Quite often if you look at the packet, it says hydrochloride, hydrochloride. So basically you're looking at the hydrochloride. That's a counterion. Right. So majority of people in the pharma industry, if they have IC in their lab, they use it to monitor the counterions. But obviously you can use it for more than just looking at those counterions. So I would say it's part of the impurities profiling in a pharma lab or even in BioPharma labs.
Right. Well, that leads nicely to my last question, which is how does IC fit into the pharma and BioPharma lab?
So I would say the way it fits in, it covers all the bits that's missing from a lot of the analysis that most people would do in a BioPharma lab, because majority of the time people have HPLCs, GCs in the lab quite readily available. But with IC, unless they know they've got counterions, then they might not have an IC in their lab. But obviously, if they did have it, they can cover a bigger range of impurities that they're looking at. And it might be something that they've not thought about looking at in their labs.
Right.
Sometimes people, sort of send it out as a contract lab to actually look at.
Right. Okay. So if people want to know more about ion chromatography, where can they get more information?
The best place to go to is our website, Thermo Fisher Scientific website, and look up ion chromatography.
Right. I'll put that link in.
Yeah, so when you go in there, there'll be sort of like various different categories you can actually look at for the impurities, for pharma, for food, for quite a lot of things, really.
Right. Okay. And have you got any webinars coming up?
Oh, gosh, loads. Talk about webinars. I have an advanced carbohydrate analysis webinar. So we are actually going to be, when I say we, I'm going to be talking about glycan analysis and about how the right element can work and simplify your carbohydrate analysis. So 2 days, 27th and 29th of April. So that's on LinkedIn if you need to sort of register. And then on top of that, we also have impurities roundtable discussion with a couple of pharma companies in May. I believe it's May the 14th. And then we've got a big global IC symposium, IC solutions, end of June.
Fantastic.
So,
well, the links for all of those will be above that. You don't have to remember anything about the links. I'll put the dates and the links above the video. Well, that's great. Well, you're going to be very busy then talking about IC over the next few weeks, then, aren't you?
Yes, well, sometimes we have to reach out as far as we can to various people.
Fantastic.
My next door neighbor is not an ion chromatography person. I believe they retired.
But I'm sure they know all about IC now, though.
They do now.
Fantastic. Well, look, that's all we've got time for. So, Wai, thank you very much for your time. It's lovely to speak to you and lovely to catch up with you.
Quite all right. Anytime if you want to chat about ion chromatography or even South London.
Definitely. And so there you go, people. If you'd like to know more about ion chromatography, as Wai just kindly mentioned, you can get in touch with her directly via LinkedIn, and leave any comments below the video, but also all the links for the various webinars and the website itself, where you've got some white papers and other documents as well, will be there as well above the video. So all this time to say thank you very much, Wai. It was lovely to see you. Thank you for watching as always. And until next time, stay well and stay safe. Bye-bye.
Thank you.